Bacterial artificial chromosomes are bags. Bacterial artificial chromosomes, they are also a type of plasmids that are used to clone very large DNA fragments. And here the size of the foreign DNA fragments may be 100,000 to 300,000. या सिंपली हम ये कह सकते हैं कि फॉरेन डीएनए का जो मैक्सिमम साइज जो कि हम बैग्स में क्लोन कर सकते हैं वो 100 से लेके 300 किलो बेस तक हो सकता है. एंड दीज bags are bacterial artificial chromosomes. They have been constructed from some natural plasmids. If you remember the F plasmids, that is important during the process of conjugation, and even its a derivative which is called as F prime that can carry the segment of bacterial chromosome, and in this situation it is called as F prime. This trait of the F plasmid has been used to construct bacterial artificial chromosomes. If we have a look on the genetic map of bacterial artificial chromosome, then we will find that it contains a PAR region from the F plasmids, and this PAR region it may be from partition, and it regulates the equal distribution of the plasmid to the daughter cells, and this PAR region it is from F plasmid. In addition to that, it also contains region of replication that can regulate the copy number of the plasmid so that it can also divide independently of the host chromosome. इसके अलावा इसमें कुछ selectable markers होते हैं, like क्लोरोफेनिकोल एंटीबायोटिक है, ये भी सिलेक्टेबल मार्कर के तौर पे इस्तेमाल होती है. और इसके अलावा जो लैक्सी जीन है जो कि बीटा ग्लैक्टोसाइडेज एंजाइम सिंथेसाइज करती है, ये भी सिलेक्टेबल मार्कर के तौर पे यूज होती है. व्हेन फॉरेन डीएनए इट इज इंसर्टेड विदिन द लैक्सी जीन, then the synthesis of the beta galactosidase it is inhibited. And this beta galactosidase that is synthesized by LAC-Z gene, it can degrade a compound which is called as 5-bromo-4-chloro-3-indolyl beta-D galactopyronosite, and simply this complex compound it is called as X-gol. When foreign DNA is not inserted within the LAC-Z gene of the bacterial artificial chromosome, then the synthesis of beta galactosidase it is there that can convert the X-gol into a blue colored end product. If blue colored colonies are there on the agar medium, it means that the LAC-Z gene it has not been inactivated. That indicates the absence of foreign DNA. On the other hand, if foreign DNA it is inserted within the LAC-Z gene portion, then beta galactosidase will not be produced, and recombinant colonies will have you can say that it will be without any blue color. So colonies will be colorless. That indicates the presence of foreign DNA. And on the other hand, if chlorophenicol it is also added, so it will also act as a selectable marker. So those bacteria will be selected that is having this bacterial artificial chromosome. When foreign DNA is inserted within bacterial artificial chromosome, then the mixture of bags and the host organism it is subjected to a process which is called as electroporation. ये cells को और जो bags का mixture है इसको expose किया जाता है electric current, और उसके बाद जो cell wall है इसमें pore generate होते हैं जिससे जो bacterial artificial chromosome है which is now recombinant, it will be you can say that inserted into the host organism. Next diagram will indicate the cloning procedure with BACS. As I told you that bacterial artificial chromosome contain a PAR region from F plasmid. It also have origin of replication, resistance genes to chloramphenicol, and cloning site that is included within the LAC-Z gene, which synthesize the enzyme beta-galactosidase. This BACS, it is digested with appropriate restriction enzyme so that cohesivents may be produced, and then the foreign DNA, it is also digested with appropriate restriction enzyme to produce cohesivent. Then they are ligated together so that recombinant bacterial artificial chromosomes they are produced that indicate the insertion of this larger DNA fragments that may be up to 300 kilobase in size. In the next step, when foreign DNA it is ligated within the bacterial artificial chromosome, then as a result of electroporation, the foreign DNA along with BACS, it is inserted into the host organism, and then they are selected on the basis of selectable marker. If the colonies they are colorless, then they will indicate the lack of beta-galactosidase, and it will also indicate the presence of foreign DNA, whereas the blue colonies they will indicate the absence of foreign DNA. So agar containing chloramphenicol and substrate for beta-galactosidase, they can act as a selectable marker. So this is one of the strategies that can be used to clone larger DNA fragments by using bacterial artificial chromosomes.